Hello everyone. In this chapter, we will address the muscular system. As we cover this chapter, I want you to keep in mind a number of questions. One, it's what are the three types of muscle tissues? Now, you remember we had a whole chapter on tissues and we addressed the muscle tissues, right? So you need to refresh your memory on that. What are the different types of muscle tissues? Two, what are the functions of the muscular system? Three, how are the skeletal muscles and the muscle fibers structured? What are the different components that make them up, right? How do the skeletal muscles contract? So we're going to dive into the physiology of exactly what is at play during muscle contraction. How do skeletal muscles obtain ATP for contraction? And what are some disorders associated with the muscular system? And how is the muscular system intervening in homeostasis, maintaining a relative internal balance, right? So these are some questions you want to keep in mind. So first up, the three types of muscles. Now, you know from many chapters back that we covered the muscle tissues, and we said that there are really three different types of muscle tissues. You have the skeletal muscle tissues, and these muscle tissues are called skeletal because typically these muscles are attached to the skeletal system, right? What is characteristic of this skeletal muscle tissue? One, they are striated. They have striations, right? As you can see, you see some light bands and dark bands, right? Two, they are going to have many nuclei in one single cell. Now, what is the mechanism by which the cell end up having many nuclei? I'm not going to go into that detail, but I want you to remember that a muscle fiber is going to have many nuclei. Okay. Also, another detail you want to know, it's that the skeletal muscles, their contraction can be controlled. You can control their contraction. Therefore, these are voluntary muscles, right? They allow for voluntary movement. You decide to walk, you decide to talk, you decide to chew, you decide to lift your arm, right? All these muscles that you control are basically skeletal muscles. The next category of muscles are the smooth muscles. The smooth muscles are these muscles that are found lie in the hollow. I mean, I'm sorry. Let me step back. The smooth muscles are found making up the walls of hollow organs. For instance, the digestive tract, right? The muscle, not the muscle, but the blood vessels, whether you're talking about arteries or veins, right? You're going to have smooth muscles. The lymphatic system, right? When you consider the lymphatic vessels, they also contain smooth muscles, right? So the smooth muscles are characterized also by the fact that they do not have any striations. They do not have any striations. Each cell has one single nucleus, right? The movement that these type of muscle does is involuntary. So you don't control. You don't control, for instance, after you swallow, the movement of the bolus the beyond your mouth, it's really actually you don't control it, right? You do not control it. In the stomach, you don't decide when the stomach is churning, right? You don't decide when the chyme is being pushed from the stomach to the duodenum, which is part of the small intestine, right? So involuntary movement. All right. So what else do I need to tell you here? I think that's about what I want to say here for the second type of muscle tissue. The last type of muscle tissue we are going to address is the cardiac muscle. Cardiac because it is found in the heart, right? Now, some characteristics of the cardiac muscle is that the cells that make up this muscle are branching, right? They are branching and they have striations and have some thick bands that appear like striations. So right here, right? These thick bands are basically called intercalated discs. In this intercalated disc, you are going to find the gap junctions. These gap junctions allow a direct communication between two neighboring cardiac muscle cells, right? Each cell is going to have one nucleus, okay? And the cardiac muscle is an involuntary muscle, so you do not decide when the heart is beating, right? The heart beats of its own, right? So involuntary movement. What is the function of the cardiac muscle? Is to pump the blood throughout the body. All right. Now let's take a look at the functions of the skeletal system. What are the functions? One is to provide support to the body. For instance, when you are in an upright position, you can stay in an upright position because the muscles maintain you in an upright position. When you are leaning, because yes, you can lean. When you are leaning sidewise or toward the front or toward the back, the muscles are the one that allow you to maintain that posture, right? The muscles allow for movement when you look at the complex motions that a dancer or an athlete can exhibit. This is made possible by muscles. But it is important to say that that kind of motion is possible because the muscles that allow this movement are attached to bones that are otherwise rigid and stiff, right? Okay? It is very important to mention that because without the bones, then it would be difficult to have this nice complex motions. Think, for instance, of a basketball player who is dribbling a ball, right? Who is running, right? So the skeletal muscles allow that. Other thing, it allows to maintain a constant body temperature because the skeletal muscles can contract, and when they contract, they produce heat. And that heat allows us to keep our inside of the body relatively warm. So the muscles also allow for the movement in the cardiac, in the cardiovascular vessels and in the lymphatic vessels. And finally, these muscles allow protection of internal organs and to stabilize joints. Now, with that being said, let's take a look at the structure of a skeletal muscle. So, a skeletal muscle is relatively complex. It's many bundles of muscles. Now, when you consider this drawing here, we have a bone, right? That's a long bone. On the bone is attached the muscle, and the muscle is attached to the bone through a tendon. And what is a tendon? A tendon, it's nothing but a dense connective tissue. Okay? So it's a dense connective tissue. Dense as opposition to loose connective tissue. Dense connective tissue that allows the skeletal muscle to attach to the bone. That's very important to know. Now, from the tendon, you are going to have going more toward the muscle, you're going to have the connective tissue that is going to form a membrane that wraps a huge bundle of muscles, right? That membrane that wraps a huge bundle of muscle is going to cover the entire muscle, right? And that muscle that is covered itself is covered before by a layer of connective tissue. That layer of connective tissue is called the fascia. Okay? The fascia is going to actually allow the bundles to be organized in many small bundles. So this is one bundle, this is another bundle, this is another bundle, right? And each of the bundle is actually called a fascicle. A fascicle itself is going to be wrapped by a layer of connective tissue, right? I'm not going to bug you with the name of that layer of connective tissue because yes, it does have a name, right? Now, that layer of connective tissue is actually wrapping a bunch of muscle fibers. So this is one muscle fiber or muscle cell. This is another muscle cell. This is another muscle cell. And inside you have the cytoskeletal fibers that are very characteristic to the skeletal muscle. These are the myofibrils. We are going to see that later, right? Now, a muscle cell is also called muscle fiber, right? A muscle cell is also called muscle fiber. Now, skeletal muscles obviously are attached to the skeletal system, right? To bones, okay? Through tendons. Now, each muscle attaches to the skeletal system at least two locations, so that one location is going to be at a bone that does not move. The other attachment, the point of attachment, is going to be on a bone that moves when the muscle contracts. So at all time, for a given skeletal muscle, we are going to have the following question: to which bone is the muscle attached, and is that bone moving or not when the muscle contracts? If the bone is not moving, then the point of attachment is going to be called the origin. Okay, let me repeat that. If the bone to which the muscle is attached is not moving, that bone where the attachment is happening, or that point of attachment is called the origin, right? Attachment of a muscle on a stationary bone, meaning the bone doesn't move during contraction of the muscle. Then you're going to have another point of attachment. That second point of attachment, it's a point of attachment that when contraction occurs, the bone actually moves. This point of attachment is called insertion. So an insertion is an attachment of a muscle on a bone that moves. All right? So to be more specific, let me show you here. So let's take a look at two sets of muscles. We're going to have the biceps muscle. So biceps brachii. So biceps brachii, when that biceps contract, what do we see? We see that the arm moves up. So the arm moves up. All right? So in other words, this attachment right here is going to cause this bone to move up when there is a contraction of the biceps. So this actually would be the insertion, right, of the biceps. While here on the bone up in the shoulder, you have some points of attachment, right, that goes on the humerus and eventually here the scapula, right? So here is the origin of this biceps brachii. Whenever the biceps contract, the shoulder doesn't move. However, the arm is going to move, right? So the shoulder is going to be the origin, the arm, and this specifically, this bone here, the radius is going to be the insertion. By the same token, when you consider triceps brachii, when triceps brachii contracts, this is going to cause a flexion, no, not a flexion, but an extension of the arm, right? That extension right here, going like this, is going to because the length of this triceps brachii is shrinking. Now, what exactly is moving here? The shoulder doesn't move, but the arm moves, right? Therefore, right here, we are going to have the insertion, okay? And this bone is the ulna, while right here, we are going to have the origin, that's the scapula, right? So, again, the tendon, which is a bundle of connective tissue, makes up the structure by which the muscle attaches to the bone. That's one. Two, the point of attachment where the bone is stationary is called the origin, while the point of attachment when the bone is actually moving is called the insertion. Please remember that. And movement occurs when we have muscle contraction. No muscle contraction, no movement. All right. So, how do we name skeletal bones? We are going to name skeletal bones based on many aspects. Some are going to be named based on the size of the muscles. For instance, we have gluteus maximus, right? That is the largest muscle on the butt, right? On the buttocks. However, you do have a smaller muscle that is gluteus medius and gluteus minimus, right? So based on the size, we name these different muscles. Then we also have a naming that is based on the shape of the muscle. Is the muscle like a triangle? Is a muscle like a diamond, right? Like a trapezius, right? So a deltoid is going to be a muscle that looks like a triangle, right? And it's called deltoid because triangle in Greek, it's delta, okay? And delta looks like a triangle. Other muscles are named based on their location. For instance, the frontalis. The frontalis is a muscle that actually attaches to the frontal bone, and that is going to be essential for facial expression. Then we do have naming that is based on the direction of the muscle fibers. For instance, rectus abdominis. Rectus abdominis is going to be longitudinally positioned on the body, right? Rectus means straight. Okay? So the naming actually can give you an idea as to the size, sometimes but not always, as to the shape, sometimes but not always, as to the location, and even the direction. And actually, well, there is something that I'm forgetting that I actually didn't mention here. By the type of movement it allows, by the type of movement, let me write it here, type of movement. When I say type of movement, for instance, some muscles do flexion. These muscles that do flexion, we call them flexors. Flexor digitorum. Digit, fingers, right? Extensor digitorum, right? They're going to extend the finger, while the flexor are going to flex the fingers, right? All right. So, other thing is the point of attachment. The brachioradialis is a muscle that is attached to the brachium and to the bone called the radius. The number of attachment. Biceps means 2. Biceps means 2. Triceps means 3, right? Triceps brachii, right? So 2 points of attachment. The action, I already mentioned that. Extensor digitorum extends the fingers. All right, so this slide here, it's a slide that actually walks you through major muscles of the human body. Now, let me tell you what I'm not going to do. I'm not going to ask you to study all these muscles and what they do. That's a lot of work, okay? I'm not going to ask you to remember all the muscles and what they do. What I want you, however, is to take a good look at this slide, look at the muscles, look at what they do, but not for the sake of the exam, all right? Now, if you are interested in, for instance, developing some more knowledge, it's very important to actually go about looking at some of these muscles and asking what is the name of the muscle, what is the action of the muscle, right? And yeah, so that would be a very relevant information to have. With that being said, I'm not going to test you on all these muscles. So let's move on. Let's focus on the muscle cell. A muscle cell of this a skeletal muscle cell is relatively complex, right? Just like any cell, you are going to have, let me write it here, you're going to have a plasma membrane. So you have a plasma membrane, all right? You're going to have a cytoplasm. You are going to have a nucleus that contains DNA, right? You are going to have a bunch of membrane-bound organelles. So now, the question I want you to ask here, it's where is the plasma membrane? Where is the plasma membrane? Well, here is the plasma membrane. But interestingly, that plasma membrane is not called plasma membrane. Rather, it's called the sarcolemma. So here is a question I want you to ask. Actually, another way to ask the question, right? It's what plays the role of the plasma membrane in a skeletal muscle cell? What plays the role of the cytoplasm in the skeletal muscle cells? How about the organelles, right? So now, with that question in mind, now you are going to know that, okay, well, the plasma membrane in a skeletal muscle cell is called the sarcolemma, okay? Now, the cytoplasm, we don't call it cytoplasm, we call it sarcoplasm. Follow me closely, right? We call it sarcoplasm, right? The nucleus is a nucleus, all right? And the organelles, well, we are going to address some of the organelles here. So, we talked about the plasma membrane. We talked about the sarcoplasm, okay? Sarcolemma and sarcoplasm. Now, let me give you some more details. The plasma membrane of the skeletal muscle cells is going to be interesting in the sense that that plasma membrane that we call the sarcolemma is actually going to fold into the cell. It's going to fold into the cell to form actually this little green stuff that you see here. So this little green stuff you see are nothing but the T tubules. The T tubules are small channels that are the extension of the sarcolemma within the cell. That is actually a little complicated to comprehend, but I want you to kind of imagine the scenario where the plasma membrane is going to form a little channel going into the cell. Now, while the channel is going into the cell, that channel does not communicate directly with the sarcoplasm. Okay? In other words, the content that is, for instance, in that channel cannot fall directly in the sarcoplasm. That is not going to happen, right? so that the outside of the cell still remains the outside of the cell in spite of the fact that the little channels are going into the cell. Hopefully you understand what I mean and you can have an idea of the picture I'm trying to show here. So you have the T tubules. Around the T tubules, you are going to have this network of purple structures right here. This purple structure, it's nothing but the endoplasmic reticulum in a muscle cell. That endoplasmic reticulum is actually the smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum in the muscle cell is not called smooth endoplasmic reticulum. Rather, it is called sarcoplasmic reticulum. Again, the smooth endoplasmic reticulum in a muscle cell is not simply called smooth endoplasmic reticulum, it is called sarcoplasmic reticulum. Okay? And that sarcoplasmic reticulum, its job is to store calcium. And calcium is essential for muscle contraction. When a muscle contracts, what actually comes into play? What comes into play, it's these myofilaments. And these myofilaments are nothing but cytoskeletal fibers, proteins, if you will, right? And these proteins are going to be actin filaments, and myosin filaments. All right? So these are the major myofibrils that you are going to find in the skeletal muscles. Okay? Now, these myofibrils, some of them are thin, some of them are thick. The actin filaments are thin, while the myosin filaments are thick. Because of that, when we look at the skeletal muscle under a microscope, we see some striations. And these striations portray dark bands and light bands. So you have light bands here, dark band here, right? Okay? And then you have some connecting point here that kind of appears dark. Okay? These connecting points are called the Z lines. Okay? Now, from one Z line to another Z line, we have this space is filled with myofilaments, and that space, it's actually called a contraction unit. That contraction unit is called a sarcomere. A sarcomere. A sarcomere, it's a contraction unit in a skeletal muscle fiber, which means that in one muscle fiber, you don't have one contraction unit, you have many of them, right? So you have from here till here, that's one contraction unit. Now, if we had more room, then we would see that, oh, there is another Z line that appears here, so that we have one contraction unit, another contraction unit, and we are not at the end of the cell yet, right? Okay, so when there is a sliding of the myofilaments, this is when we see a contraction, right? So let us move on. I know I said a bunch of things here, but please bear with me. All right, so this slide here basically summarizes all that we've said so far. right? The sarcolemma is the plasma membrane in a muscle fiber, and that plasma membrane actually folds into T tubules. Okay? We have the sarcoplasm, which is the cytoplasm of muscle fibers. We have myoglobin, or that one I did not mention that molecule. Myoglobin, it's a type of pigment, it's a red pigment, right? That actually carries oxygen in our muscles, and oxygen is essential for what? For cellular respiration, right? And what is cellular respiration? Is the cellular process by which we convert the chemical energy in, for instance, glucose into ATP, which is the currency of energy that is necessary for accomplishing cellular work, right? Then we have the T tubules. The T tubules, an extension of the sarcolemma that extend into the muscle fibers, right? And it plays a very complex role. And the very specific complex role is actually to convey the information that comes from outside toward inside so that contraction can happen. More specifically, you have calcium that can be released by the sarcoplasmic reticulum because of the signal that is coming from outside. But that's another story that we are going to talk about later. Then we have the sarcoplasmic reticulum. The sarcoplasmic reticulum, it's a smooth endoplasmic reticulum, right? Its job is to store calcium. That very calcium is going to be essential for muscle contraction. The myofibrils, these are bundles of myofilaments that are essential for contraction. And the myofilaments are going to be actin filaments and myosin filaments that are going to work together for muscle contraction to occur. So now, let's zoom into the sarcomere. When we zoom into the sarcomere, we find that there are two major types of protein occurring in there. We have thin proteins of actins, okay, making up thin filaments, and we have thick proteins making up myosin or thick filaments, right? Now, the structure is very complex, but I'm going to spare you details. Here is what I want you to remember. The actin filaments are made up of actin proteins, and these actin proteins, as they organize filaments, they are actually two strands that are twisted around each other, kind of like DNA, but don't go into the details of that. Let's not go into the details of that. So we have two strands of actin filaments that are connected together, that are wrapped around each other to form a nice filament that is stronger than one single filament, right? So that's that. The second thing I need to mention, it's the myosin. The myosin, a single protein of myosin, it's kind of organized like a golf club, right? It's kind of organized like this. Okay? So you're going to have the head. There we go. So you're going to have, let me change some color here. So you're going to have here what we call the myosin head, right? And the remainder of the body, it's basically a filament that allows to connect, to intertwine with other myosin molecules. All right? So now you understand that these are a bunch of myosin heads that are here, and bunch of myosin heads that are here. So you will see when you look at a sarcomere, you have some myosin head, some myosin protein that go this direction, while others that go this direction, right? And then you are going to have actin filaments in here and actin filament in here, as if the heads of myosins could attach to actin filaments. In fact, they do. Okay? They do. And that attachment and change in the conformation of the head is what allows contraction. Okay? So, now the Z line, it's actually where actin filaments are attached to each other. Now, let me readjust what I just said. So the Z line, it's actin filaments from one sarcomere are going to attach to actin filament from another sarcomere. In other words, this is sarcomere 1 and this is sarcomere 2 right here. So this actin filament belongs to this sarcomere right here, while this belongs to this other sarcomere right here, right? So that's the Z line, okay? Now, here though, you have the myosin filaments that belong to the same sarcomere that are going to become intertwined here, 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 all right? So, I want you to keep that in mind. So, with that said, another dimension is that now when we look at a sarcomere under a microscope, what do we see? Now, we see a Z line right here and a Z line right here. So that makes it from one Z line to another Z line, we are talking about a sarcomere here. Then in between, what do we see? We see this part that looks dark under a microscope. This is what we call the A band. The A band is the darkest part in the striations that you see. Then you have a light part. A light part, it's actually going from here, right, to here, where all the myosin filaments are going to appear here. That light part, it's actually the I band, okay? And that I band is light because actually you have a bunch of space, empty spots here. So light has a chance to cross here more than it can cross here because of how much this area is cluttered, right? All right. So then, out of the A band, you have a particular area right here that we call the H band. That H band is slightly light compared to the remainder of the A band. Okay? So that's basically all the little things that are present in a sarcomere. So the Z line, the A band, the I band. Now, let me start over. From one Z line to one Z line, we have a sarcomere. An A band, it's the dark band. An I band is the light band. An H band, it's the middle of the A band. All right? And now, so this sarcomere here is relaxed, while this sarcomere here is contracted. Okay? You can see how the H band has disappeared in the contracted sarcomere, right? The A band has disappeared. Why? Because this actin filament moved this direction, and this one moved this direction so that they came closer together, right? That's contraction. All right. So now, what happens during muscle contraction? Okay, this is going to be a little bit complex, but please follow along with me. So, first and foremost, there is no muscle contraction without stimulation. That is something you have to know upfront. The question you need to ask now is, wait a minute, what is causing that contraction? What is triggering that contraction? What is the stimulus? Well, the nervous tissue right here carries the impulse, and that impulse is going to travel through the axon and eventually reach the synapse, okay? The axon terminal. That axon terminal is going to end up with a button that we call a synapse button, and we are going to actually have what we call a neuromuscular junction. That neuromuscular junction, neuro for neuron, muscular muscle, junction for the connecting point, right? So here we have a zoom in that neuromuscular junction, okay? In that neuromuscular junction, what exactly is happening here? Well, we are going to have that at the synaptic button. What do we have? We have some mitochondria. Mitochondria are there to produce ATP. ATP is essential for action, right? We have some vesicles. These vesicles contain some green particles, and those green particles are called neurotransmitters. Neurotransmitters. These neurotransmitters, in this particular instance, are called acetylcholine. Acetylcholine, right? Acetylcholine, right here. It's abbreviated ACH, acetylcholine. Now, acetylcholine is released in the space between the synapse, right here, and the skeletal muscle, right? So you have a space that is called the synaptic cleft, okay? That synaptic cleft is where the neurotransmitters are going to be released. Now, let's zoom in that neuromuscular junction. So ATP causes the release of acetylcholine in the synaptic cleft by a process we call exocytosis. Okay? Now, the neurotransmitter is in the synaptic cleft. That neurotransmitter is going to target the receptors right here. Now, the neurotransmitter is called acetylcholine. The receptor for acetylcholine are called acetylcholine receptors, right? That binding of acetylcholine on acetylcholine receptors is going to send the signal inside of the muscle cell, right? As the information flows inside of the muscle cell, right, by way of cell communication, the sarcoplasm, the sarcoplasmic reticulum, will release calcium. That calcium is now going to go and bind on some proteins in the myofibrils. That is the next part of muscle contraction that I'm not going to address right here, but in the next slide. Okay, let us move on. So now, remember that the impulse has come, right, through the axon, reaches the axon terminal. A transport vesicle is going to release a neurotransmitter, in this case acetylcholine. Acetylcholine gets in the synaptic cleft, and there acetylcholine is going to bind on acetylcholine receptor, which receptor are embedded in the plasma membrane of the muscle cell, right? So in the sarcoplasm, right? Okay, a sarcolemma, I'm sorry, sarcolemma, right? So the receptors now activated is going to send a signal inside of the muscle cell to cause a release of calcium. Now, calcium is released by the sarcoplasmic reticulum, and where is that calcium going? That calcium is going and binding on an accessory protein. That accessory protein is called troponin. Upon the formation of the complex troponin calcium, troponin is going to change its conformation. By changing its conformation, this is going to cause tropomyosin to slide. And by sliding off, actually what happens is that tropomyosin, right, the green protein here, is going to free these spots on the actin proteins. All right? These spots are actually binding sites. They are binding sites for what? They are binding sites for the myosin heads. Remember that myosin heads, all right? Myosin, it's kind of like a club, right? Okay? So this club is going to attach to an area. Oops, no, that's not like this, okay? So there is a little spot right here where connection is going to happen, right? So now you know. And now we are going to zoom in. When we zoom in, so first, remember, we need ATP. So ATP is going to come into play. ATP is going to come and attach to the myosin head. By attaching to the myosin head, that ATP is going to split. It's going to split into ADP on this side and a phosphate on this side. Okay? That binding of ATP on the myosin head is going to actually cause an attachment of the myosin head on the myosin binding site, forming what we call a cross bridge. Now, that cross bridge is going to exist in as much as phosphate and ADP are attached to the myosin head. Then something is going to happen. That something is actually hydrolysis of ATP. That hydrolysis of ATP, it's simply that an ADP molecule is going to remove from the head, and the phosphate group is going to move away from the head. When that happens, we are right here. When that happens, this is going to cause a cocking or a stroke of the myosin head, causing the myosin head to move this direction. That movement, it's actually what causes the myosin to slide alongside the actin, so that the actin going to go this way, right? Okay? While the myosin heads are going to move this direction, right? Same direction, really. Okay? Now, what you need to imagine is that what is happening with this myosin head is also happening somewhere else, right? It's also happening somewhere else. So now we have a cocking here. And eventually, that position, that is the contraction position, the contracting position, will have to be released for a muscle to relax. What caused that relaxation? Well, ATP is going to be invested. That ATP is going to come and bind on the myosin head. Upon binding, the myosin head is going to go back to its original position. Okay? So, the binding of ATP on the myosin head causes the myosin head to go in its opposite, its initial position, while a dissociation of ATP from the myosin head causes the myosin head to actually move in one particular direction. All right? So that's muscle contraction. And actually, what you see here, it's basically, you can imagine that the hands right here represents the myosin head. And the myosin head, as they are pulling, what are they pulling? They are actually pulling the actin filaments. All right? So that you can have an idea here. All right. So now, let's go back. The nerves are going to release some nerve impulse, right? They travel down a motor neuron to release a neurotransmitter at a neuromuscular junction. The neurotransmitter here, it's acetylcholine. When acetylcholine is released, it's going to go and bind on the sarcolemma, and specifically on the receptors, right? When acetylcholine binds on the acetylcholine receptor, right, the information goes inside of the cell, and the sarcoplasmic reticulum is going to release calcium. And that calcium is going to go and bind on troponin. Upon binding of calcium on troponin, troponin is going to change its conformation, which is actually going to affect the positioning of tropomyosin, which frees the myosin binding sites, right? And now the myosin heads can form a cross bridge with the myosin binding sites. ATP binds to the head, right? A split of the ATP molecule is going to cause the binding of the head on, well, actually, it's going to cause actually a release of the, not a release, I'm sorry. It's going to cause a cocking of the myosin head, right? Shifts, right? So this is going to actually cause a sarcomere to contract, right? And then as ATP attaches to the myosin is going to actually cock. And when ATP comes back and attached to the myosin head is going to come back to its normal position. All right? So that we know. Now, what we also need to know is that after death, because there were still some ATP in the muscles at the time of the death, actually that ATP was used for muscle contraction, right? Eventually, muscle cells are going to continue to produce ATP, not by aerobic cellular respiration, but by fermentation, okay? And eventually, there is going to reach a point when the cell is not going to be able to do fermentation anymore. All right? Because the system is not maintained, right? So when ATP runs out, now some myosin heads are going to still be attached to the actin filaments, causing this sarcomere to remain shortened. This situation is called rigor mortis. That rigor mortis allows investigators to determine exactly how long ago a person died. Okay? So rigor mortis, along with the temperature of the body, allows investigators to estimate the time of death, right? So here is an example of someone who was discovered died, and the rigor mortis along with temperature were used to actually estimate the time of death for that individual. So basically, this is muscle contraction. For now, I'm going to stop here. Thank you for your attention.